Hello, my name is Charles Blatti, and I'm a research scientist at the National Center for Supercomputing Applications at the University of Illinois. Today I will present to you a recent tool we have been developing called PhyloDiver. Before I begin, some background information. Our tool has been developed for the field of cancer phylogenetics. The basic tenet of this field is that cancer is the result of an evolutionary process. Mutations occur and accumulate in a population of cells through time as the cells divide. The tumor, therefore, is composed of multiple subpopulations of cells, or subclones, each with distinct patterns of mutations. This intra-tumor heterogeneity may drive a cancer's rate of progression or its resistance to therapy. Researchers use evolutionary trees, or phylogenies, to better understand this heterogeneity in their tumor sequencing data. In order to produce and interpret these phylogenies, a many-stepped analysis workflow is required. First, bulk sequencing data should be sent through a best practices somatic variant caller. Then those variants need to be annotated, and then to produce the phylogeny, the variants need to be grouped into clusters, and then the evolutionary tree is built by determining the relationships between those clusters. Finally, additional information, such as known mutational signatures, can be overlaid on the resulting tree. For each of these workflow steps, there are several different outputs, here denoted in the colored text of each step. So cancer phylogenetic researchers need to understand the history of tumor evolution and the clonal composition of the samples from a patient. Therefore, any visualization tools should be designed to answer these core questions. The tools should allow the researcher to access the different result outputs from each workflow step. There are many existing tools for visualizing tumor phylogeny, but they typically fall within two categories. The first type of tool often has a static image of clonal evolution that does not accommodate user interaction. These images are saved at the end of the tree reconstruction step and provide an overview of the evolutionary relationships between the subclones and the cellular proportions across the related samples. These images are often called fish or bell plots and are produced by the tools listed here. Because they are static images, they typically do not integrate details about the mutation events or their annotations into these visual histories. The second type of visualization tool frequently used is the interactive tree layout interfaces. These tools excel at enabling tree-based exploration and can even accommodate some level of annotation added to the branches or to the nodes. However, these tools lack that area-filling visual aspect of the fish plots that conveys information about the cellular proportion of the subclones. This stems from the fact that most of these example tools of this type are generalized phylogeny viewers, more designed with gene or species evolution in mind. So we built PhyloDiver, a new cancer-specific visualization interface that attempts to preserve the best features of both types of existing visualization tools. The purpose of PhyloDiver is to better enable end users to examine the clonal evolution in an interactive fashion while still remaining connected to the underlying mutations and annotations from all the steps of the analysis workflow. So let's take a look at some of PhyloDiver's features and actions. First, we developed a tree visualization that always remains tied to the cellular proportions of the selected subclones. Here you see our demo with deep sequencing data from an AML primary tumor and its subsequent relapse. There are 6 subclonal populations across both samples, represented by the different colors. When we select the primary tumor, the proportions are listed and sized accordingly, and so too are the corresponding leaves of the tree. When we select a subclone, the tree browser zooms in and we see the accumulated mutations of that subclone. And then we can also switch to the relapse sample, and again the tree resizes to match the new proportions. The next set of PhyloDiver features are tied to its summary of mutation clusters. When we click on an edge, we see a summary of all the mutations that were acquired at that phase of tumor evolution and some of the information about their related annotations. In this example, when we look at the most recent mutations found in the yellow subclone, we see that only one of 354 mutations found in this cluster are predicted to have a high impact based on its consequence from the variant effect predictor. We also see that these mutations occurred in 202 unique genes, and 9 of which are known cancer genes according to the Cancer Gene Census. Finally, using information from the GDI database, we can see that 44 of the genes are known to interact with different drugs and small molecules, information that can potentially impact treatment decisions for this patient. Besides seeing the information for the selected edge, we also see a mini summary for the other edges of the tree, which help the user to select compelling mutation clusters to follow up on. Finally, we show that the PhyloDiver visualization is tied to the underlying mutation details. Again, if we select a specific mutation cluster, we can look at all of its variants of a predicted severity by double-clicking. This opens the detailed table of that cluster, and the severity filter is applied. If we want to instead see all the subclones for that severity, if we want to see all the mutations for that severity for the related subclone, we can still use the navigator above. We can also remove the filter on severity to see the table of all the mutations for that subclone. PhyloDiver is preloaded with 2 demo data sets for you to explore. This AML one and another demo using samples from a patient with colorectal cancer. We also support the uploading of user data. When you select to upload your own data, it is loaded locally in your browser. The upload consists of a single file following our custom JSON specification. We have documentation and tools for reformatting the data yourself, but we also have an alternative option. Our PhyloFlow workflow is an end-to-end cancer phylogenetic analysis starting from the VCF inputs and running through the specific tools for the remaining steps. The final command of this workflow merges all the individual step outputs into the PhyloDiver file with the correct JSON format. This workflow is containerized and has a WDL workflow description, and you can check out the public version of PhyloFlow at the doc store, where you can launch it on various cloud platforms. There are many talented people to thank for their efforts on this project, and we're especially grateful to the Cancer Center at Illinois who provided funding. And then finally, in summary, our new PhyloDiver tool enables researchers to interactively visualize and explore a multi-sample tumor phylogeny. This tool, along with our analysis workflow, represents the next step in promoting broader adoption to the methods of cancer phylogenetics for understanding tumor genesis, metastasis, and drug resistance. Thank you. Are there any questions?